and thank you for everybody for being here today. It's a great opportunity to talk about some of the collaborative projects that we have ongoing. This is a big focus of our lab here at NINR, as much of the collaborations really exemplify our ability to pair our expertise here at NINR, both in the clinic as well as in the lab, with experts here on campus as well as around the world. And what I've found through these types of collaborations is it allows us to develop fundamental insights into the mechanisms underlying the onset of deficits and symptoms following brain injuries, and really to do science that's collaborative and moves the efforts forward to understand the comprehensive mechanisms as well as the consequences of brain injuries. And so today, I'll talk about one of these collaborations, specifically that we have with the Department of Defense to look at blast biomarkers in the blood. And in this collaboration, we focused on blast with the understanding that blast is one of the clinical injuries most often occurred in recent deployments in Iraq and Afghanistan. We now have unprecedented numbers of military personnel and veterans that have gone on to have long-lasting symptoms, both neurological as well as behavioral, following these blast exposures. This leads our group, as well as others, to question if we can use biomarkers to understand the mechanisms underlying these symptoms and deficits. But this is difficult, as many of these individuals have these blast exposures during deployments that are in remote combat stations. Also, when individuals are exposed to blasts, they often have blunt force traumatic brain injuries. What we know from preclinical models is blunt force injuries are very different in their neurological sequelae from that of blast. So really the focus of this collaboration is to look at blood-based biomarkers, and specifically those that are unique to blast exposure, and to bring this research here to NIH. So to do this, we partnered with military collaborators at the Department of Defense, and specifically at Walter Reed, and we were able to work with service branches of the Army as well as the Navy. And those investigators were experts in both blast measurement as well as giving us access to populations to be able to look at training-related blast exposures. This gives us two unique possibilities. First, we're able to collect baseline samples on individuals prior to any type of blast exposure. What we know from our studies in the lab is that every individual has a very different proteomic activity as well as genomic activity. So by having this baseline level, we're able to look at individual variations and really increase the sensitivity of the assays in our lab. In addition, we're able to use blast measurements that are approximated through sensors that are within the helmets of these individuals. So we're actually able to look at blast exposure proximity as well as the overall severity of that blast. We're able to collect blood in these training experiences and to add our blood collection protocols to these existing protocols to allow blood-based biomarkers in these individuals. Lastly, we're also able to look at the exclusive impact of blast, as that these individuals very rarely will have any type of blunt force brain injury. So again, we're able to highlight the specific components of blast and look at these biomarkers in these individuals. So we paired the expertise of our military collaborators who have access to these training samples, populations, and also know how to actually measure this blast and approximate the severity, as well as other blast parameters that are important to measure when we're looking at biomarkers. We paired that with our expertise in the lab to be able to do accurate and in-depth phenotyping of these biological markers, as well as to pair them with the clinical implications that they may result in. We looked at a myriad of different biomarker profiles, including genomic expression, but today what I'm going to talk about are some of the protein studies. Specifically, we use an ultrasensitive assay that allows us to look at very small concentrations of proteins in the blood that we know relate to brain injury. And specifically today, I'll talk about our studies looking at amyloid beta, as well as tau and phosphorylated tau. We paired with our military collaborators to do this study in which individuals were engaged in a 10-day training period. We collected blood at both baseline as well as every day during training. So these individuals were engaged in blast-related training and had these sensors that approximated their blast exposure, and then we were able to collect blood between the hours of 4 and 6 p.m. on all of these training days, allowing us again to look at individual change over time in these individuals, looking at these biomarkers. We compared cases who had a moderate blast exposure on day 7, which was indicated by a PSI of over 5. To give a context to that, anything over 7 is where an individual can have the bursting of their eardrums. So this is a substantial blast exposure. We compared these individuals to controls who were also engaged in training but did not have any blast exposures over that of 2 psi. And what we found was something really surprising. We found that overall, there were reductions in amyloid beta 40 and 42, as well as tau following blast exposure. This is counterintuitive to all the other studies that we've done in our lab, where we've looked at civilians with mild and moderate brain injuries, as well as athletes with concussions. We consistently see increases in these three proteins. In addition, we see increases in phosphorylated tau. This is problematic, as we know that when tau becomes hyperphosphorylated, it's much more likely to result in neuronal aggregations, resulting in the form of tau tangles, which have been implicated in chronic traumatic encephalopathy, or CTE, in which individuals will have a concussion or multiple concussions and will go on to have this dementia-like presentation. In addition, we also looked at ratios of these proteins, finding that the ratio of amyloid beta 40 and 42, as well as tau and amyloid beta 42, were significantly altered after blast. This is particularly concerning, as these are two of the most predictive biomarkers for individuals with mild cognitive impairment to then go on to progress to Alzheimer's disease. Again, we're seeing an alteration in the phosphorylation of tau following blast, again, suggesting that there may be some neurodegenerative pathology that's initiated by this blast exposure. Putting these findings together, first, by pairing our expertise in the lab, by running these assays over and over, we know that we're seeing something very different in blast-related biomarkers. This will be the first study to show that blast biomarkers are different from blunt force, really leading to clinical insights that have implications in military personnel and veterans. In addition, we're also seeing that some of these suggest neurodegenerative pathology, leading us to do additional clinical studies here at NIH, as well as at other military sites, to pair these neuroimaging changes with these biomarkers to understand comprehensively how blasts may relate in neuronal pathology and may suggestively relate to chronic symptoms and deficits. Lastly, we're pairing with RARE also to do preclinical studies to understand the mechanisms underlying these clinical observations, and specifically to look at the lymphatic system. Specifically, we look at this system as we know that lymphatic flow is essential for the clearance of tau as well as amyloid beta following a brain injury. So this may be the mechanism that underlies some of our observations and also gives us a place to intervene. So by pairing our expertise here at NINR with our military collaborators, we're really able to move this science forward and understand the unique components of blast to give some therapeutic insights in how to address this and improve the care of military personnel and veterans. And with that, I'll thank my lab as well as our collaborators who make this research possible. Thank you.
Thank you for having me here this afternoon and for your attention now. What I'd like to talk to you about today are the brain-gut connections and collaborations that we've been able to forge and continue to do, as well as updates from last year when we were able to present to you. So as Dr. Cashion spoke earlier about the Symptom Science Model and Dr. Saligan about our Symptom Science Center, this is our mission. This is our focused effort as to how to help our patients, help them with the symptoms they have, but more so in doing so, to phenotype these complex symptoms that our patients tell us about, specifically characterize these, look for biomarker discovery from an innovative perspective, and then moving on to clinical application, wherein the intent is, with these applications, we will improve their symptoms. So the focus of my lab is digestive disorders. So these are many and varied, and as we know, stress affects GI disorders across the lifespan. So specifically, I'm interested in the brain-gut microbiota interplay and how chronic stress affects intestinal health across the lifespan. Up to 20% of the US population reports stress-induced symptoms. It's the single most common reason for emergency room visits, top 10 for outpatient visits, 30 billion costs annually, and there is evidence that chronic stress early in life or at any point in life affects health across the lifespan. So if you remember our symptom science model, now imagine that operationalized from my stress-induced perspective. So what we've been able to do, and we'll move through this, is to be able to phenotype through innovation. So this here is called the gastrointestinal pain pointer that we developed in-house in the clinical center at NIH. And it allows patients to tell us where it hurts, dial up how much it hurts, use word descriptors, and also get measures of the sympathetic and autonomic function with heart rate and blood pressure in real time. So that was the first thing. If you're going to phenotype and ask questions of how your patients feel and their symptoms, we had to have really good measures. And those measures, to be able to validate that in this environment, has been invaluable. So that was the first sort of step in our program. Then what patients do within our cohorts is we ask them about their GI discomfort with the GI pain pointer, and then we actually try to recapitulate their symptoms. And we do that by a metabolic stressor. So it's a combination of sugars called the brain gut test solution. It's a 100 ml test solution seen here. Patients ingest this orally in a fasting state in the clinical center, and then we measure the metabolism of those sugars across the GI tract over a 5-hour period. We measure this via mass spec, and we have a highly quantitative way of doing this that we developed in collaboration with the Department of Laboratory Medicine, and now a new, more highly quantitative way with NIDDK. So that's very exciting that we have this measure. It allows for different sugars that permeate with different molar masses at different points of the GI tract to be able to identify altered permeability. We then move on to our personalized profile. So, okay, we've been able to recapitulate their symptoms. They've told us where it hurts, how it hurts. We know at what point in time they have this altered permeability. But what else goes with this? What is this gene environment? And how's the brain and the gut and the microbiota working together? So we've been able to publish some of these results as well, and worked with lots of help here at NIH, along with some extramural collaborators and companies to validate this genomic data to ensure that indeed it is reproducible. So on to clinical applications. What we've been developing is actually a gold nanoparticle lateral flow assay that we developed at the bench here at NINR, which has been my dream as a nurse practitioner when a baby or a patient comes to me and they have symptoms, especially of diarrhea, that I know what to do before they leave me and go back to the community and can't ever get to me again. And so we've been able to develop that, and I'll talk to you about that. But specifically, I want to give you some examples. So we have ongoing studies. So we have our own natural history protocol that I have a co-PI, Dr. Margaret Heitkamper from University of Washington, who's also one of my mentors extraordinaire, with over 130 patients enrolled that have had this protocol of the brain gut test solution, the GI pain pointer. And we also have a natural history protocol where we do indeed deliver care to patients. Patients can come back to our clinic. It's also a training protocol for our postdoctoral fellows and nurse practitioners to be able to treat patients with standard of care. And then the protocol specifically, and you can read on to these other collaborations, that I want to highlight is a collaboration with Mass General and Harvard with Dr. Janet Lowe. You can see a few other of the intramural collaborations, as well as some bench-to-bedside funding that we've collaborated with NIAID, and some other mechanisms and funding for the fellows in the lab. So this particular study is entitled Targeting GI Epithelial Integrity to Improve Arterial Inflammation in HIV. And really, to kind of walk you through the steps to look at sort of a picture, is the idea that if you have chronic stress effects, and that you have altered GI permeability or GI integrity. So in this study, we look specifically at this via upper and lower endoscopies and biopsies, that you'll have microbial translocation that'll be ignited. So basically microbes moving across the GI tract. And that this translocation of these microbes activates the immune system, which then that chronic inflammatory immune state may be, and this is a high-risk hypothesis, related to atherosclerosis and chronic inflammation from a cardiovascular standpoint. So in this particular protocol, it's an HIV protocol where patients, as well as healthy controls, and I'll walk you through the algorithm, actually have an upper and lower endoscopy. They get tissue biopsies. We assess for tight junctions and altered permeability, as well as apoptosis. We characterize the tissue-specific T cells and macrophages. We look at these measures via blood, as well as biopsies. We assess intestinal permeability, so our own NINR methods of the GI pain pointer and the brain gut test solution are given to these individuals before randomization to drug and then thereafter. We also assess the microbiome. So just quickly in terms of study design, patients come in, they are screened, they have intestinal permeability as well as DEXA scans and other markers that go along with inflammation and cardiovascular abnormalities. Because the drug specifically is a GLP-2 medication that's injected directly into the abdomen and regenerates the gut epithelium specifically, if you have any lumps, bumps, nooks, or crannies whatsoever, they will proliferate. So patients have to have fully clean upper and lower endoscopies before they go on drug. So at any rate, we also have healthy controls for comparison, and we've completed we're now up to 9 patients who have healthy controls that have completed that baseline evaluation. And then patients that are living with HIV are randomized to either placebo or drug, and then they have 6 months of treatment with GLP-2. And so there's safety visits, and it's a daily injection into the abdomen of teglitide, which is GLP-2, or placebo for 6 months. Then they have the follow-up cardiovascular evaluation, the PET-FDG, as well as the intestinal permeability, DEXA scans, and so forth, and then repeat upper and lower endoscopies. So that's an example of a collaboration, an example of other institutes utilizing some of the innovation directly out of NINR. And then in terms of future studies and ongoing work, from our diarrhea symptom perspective, of course, patients with HIV, as well as many patients today, given our current environmental state that we have, where we've had hurricanes and so forth, and there may be disease coming, we have developed this lateral flow assay through a CRADA collaboration, which is a clinical research and development collaboration with an outside entity called GoDX. And this basically allows for gold nanoparticle labeling of direct synthetic nucleic acid detection without electricity, without any reagents, and that can be detected with the naked eye. So last year I talked to you about this. I gave you a few comments about it. Well, it's since been funded through SBIR to GoDX, our collaborator, by NCATS for phase 1. We're almost complete with phase 1, and now we're moving on to applying for phase 2. And also for a clinical protocol, potentially. We've made it through scientific review in-house at NIH, where we'll actually test these tools on patients in-house in real time. So we know it works on the bench with synthetics and with healthy biofluids from humans, but next is the bigger study of over 800 patient samples to be able to validate the sensitivity and specificity in real time. So we're looking for lots of collaborations because we need lots of samples, but more so than that, we're just very, very lucky to have this opportunity that NINR has given us, and to have collaborators both within NIH as well as outside of NIH, and we're looking forward to other partners in the very near future to be able to help patients and to be able to do what we do best, which is to assess their symptoms and do something about it quickly. Thank you.
I think, again, our presenters have shown the breadth and the depth of our collaborations. I would like to, before I ask the first question, actually acknowledge a question that I frequently get is, are we just members of the team at the table? And I would like to say that I think that y'all have shown very clearly that you're the leaders of the team, and you are pushing the science forward as nursing scientists. So, and you've talked about those collaborations. Again, you talked about successful collaborations. So I'd like to ask you the same question I asked our first 2 panelists. Is related to, in your successful collaboration, there are always challenges. Could you maybe discuss one of the challenges or a barrier that you overcame successfully and you've moved forward?
So I can go first. I think one of the barriers, especially when we're working with different federal agencies, is how to get the regulations approved through all of our IRBs, how to make sure that everything's in agreement so that way we can do the science. I find that there's a year worth of paperwork, then we actually do the science. So a lot of that has been learning how to really expedite things and to make sure that they go onto the desk of the right person, and to be assertive enough to make sure that those protocols move forward, because we can't do this work unless we get the regulations right, and we really need that. And we need cooperation from different federal agencies as well as the military to do that work. So that's been one of the challenges that I've faced.
Dr. Henderson?
I think this is probably a common challenge across probably all clinicians and investigators and research, is being able to validate your work in other settings and have other individuals validate your work, and to come together on these common data elements. And I think what's been really great is that NINR has been really at the forefront of developing, who and what and when. And yes, you may have other ways to evaluate patients and particularly phenotype, but from a symptom perspective, if there's a way we can come together at the table and have a common data or a common element or a common group of patients, I think it's really helpful. And I think moving that forward has been huge, especially towards collaborations in the future.
Thank you. So, Dr. Henderson, could you tell us a little bit about how your experience as a nurse practitioner has actually prepared you to lead a collaborative research team?
That's a great question. Actually, it was my experience as a nurse first. So I was a recruitment coordinator for the diabetes prevention program back in 94, 95, trained by Matthews Media and NIH through NIDDK to go around to 25 centers across the United States to train in standard outcomes. And I think that experience and that standardized experience with standard data cores, but then going into different Indian reservations, going into different communities where the way you do things doesn't make any sense and may even violate that culture, and to have some of that understanding and bring it back, and then for it to be heard as a nurse, and then for someone to say, hey, you need to go back to school and stay in school and let's make a bigger impact, let's keep going. And I think if we can continue to encourage the next generation of nurse scientists to have that next seat at the table, and when we're ready to back away from the table, someone else is ready to sit down, I think that'd be huge.
Thank you. Dr. Gill, what challenges and successes have you experienced in your collaboration with military scientists through the Center for Neuroscience and Regenerative Medicine?
Absolutely. So the Center for Neuroscience and Regenerative Medicine was initiated in 2007 here on campus to link our ability to do clinical research here at NIH with investigators over at Walter Reed, as well as Uniformed Services University of the Health Sciences. Again, one of the major things that we've had is how can we get common IRBs to really understand the science that we're doing. Every time we do any type of amendment, we have to go through all of the IRBs. That can be cumbersome. But also kind of the thing of all of the scientists coming together really to motivate the science, because that's how we can do it. And so we've set up a common biomarker core so that way we can do that type of science, as well as a neuroimaging core, informatics core. So that way all of us scientists can work together. So that's been one of the major advantages of those years of paperwork to get that done. But it's a constant process of making sure that we're doing the right thing, but we're also progressing science as much forward as we can.
I'd like to open it up to questions from council. Shirley, go ahead.
When I think of the large symptom science initiative for nursing, I guess my question's involving the collaboration between the internal and the external programs. When I see what's happening in the external programs, intervention testing, all right, and it seems like your model of complex symptoms, symptom characterization, biomarker discovery, clinical application, I'm assuming the intervention testing comes under clinical, but I haven't heard much about, are you expecting that the external people will come up with the possible interventions then based on your discoveries at the other two levels? I'm just wondering how we can collaborate more. We're testing gobs of interventions, and I'm not sure we're bringing in your, and maybe it's a stage thing, we're not there yet, but.
No, I think you're absolutely there. You're where you need to be. And that's, 95+% of our budget. So if the interventions can be done extramurally, they should be done. So for example, with my study, I took what we developed here, and then I went and taught the nurses in the GCRC, at Harvard and Mass General how to do that, and then apply another novel intervention. So really the intent is not for us to do everything, it's for us, and this is my understanding, correct me if I'm wrong, but to come up with novelty and so forth. It's taxpayers' dollar. It belongs to the nation and the people, and it really should be given back. And the intent is not for us to do everything, for us to sort of kind of get it going and then give it back.
So you don't expect to see yourselves doing randomized trials of interventions, large ones?
Go ahead.
So that's part of my program of research based on the mechanisms we see, but we definitely collaborate with extramural investigators to do multi-site trials. I think that's really where we're going with brain injury research, because that's where we really can get the maximized benefit to do that. Here at NIH, we can do in-depth phenotyping in ways that we can't do other places, so we maximize our unique abilities here and then try to pair it with extramural collaborators to really maximize the benefit of the research we do.
And I'll just add one piece to that. As the director of the program, what I really want to see is the science move forward, whether that's we are doing more phenotypic characterization or looking more at the interventions. It's not one or the other, it's which one can we do in a timely manner and make a difference. So that's why you hear we do a lot in all the boxes right now. Go ahead, yes.
So to all four of you, your programs are so impressive. Congratulations. A lot of the protocols that you have are really intense for the subject. And so I wonder if you can talk about how you keep your subjects engaged in going through all of that testing.
Well, honestly, in some of the protocols, especially the collaborative protocols where other types of rare disorders come in and do our brain gut protocol, participants know that we may not actually help them, but we may be able to generate or better understand what's going on with them for their next generation or for others. And so I think, the real part of it, yes, there's motivation in being able to compensate someone financially, potentially, although they don't really get compensated as well intramurally as I think extramurally. But the other part is really there's this altruistic mentality, and people really want to come here, and they really want to contribute and give of their time. And so, yes, we feed them after we fasted them, and yes, they get, but they also get some of their results too. Some they don't. But I think that there's motivation there, especially if they present with symptoms. If there's the potential to learn from that, it could be helpful otherwise. And at least for my cohorts of patients, we don't understand and we have no treatment for their symptoms. And so they really are looking for help.
Thanks. I echo the impressive nature of the work that's going on here, and I have a new appreciation, I think, today of what is actually going on here. And I don't have so much a question as sort of a comment and a request that somehow these talks this afternoon, these questions and answers and give and take, somehow get communicated as well to our extramural community around this collaboration and the leadership and participation and really true collaboration across disciplines, which is very difficult, I think, and you all certainly are representing that quite well. And our folks out there, particularly our young folks, need to hear it.
Thank you, Nancy.
I wanted to echo what others said. I think particularly what Shirley said. I think that all your collaborations are very impressive with intramurally as well as with extramural people, and it's very obvious. And I mean, in a smaller way, I've done some of this, so I understand how much work and appreciate your comments about the IRB. But I really, it feels like, this program has grown a lot intramurally. You guys have grown a lot professionally. And then some of us, as was pointed out, are doing clinical trials on symptoms out in the field, funded by the NINR's extramural program. And I would really like to see us as an institute find a few, you've done it selectively for your own specific interests, and that's great. But is there a way that we could think about a mechanism institutionally going forward to partner extramural people with intramural people to push the basic science out into clinical trial and then to dissemination? Is there a way to do a program announcement, or is there a way, to kind of, I don't know what the mechanism would be, but I think it might be the time to start to plan for something like that.
And, we'd really love to be able to do that. In fact, Anna and I have talked about that a bit to figure out what the timing is and what do we have to offer and how do we set that up. We're very open to suggestions as to how that might take place. We weren't, a few years ago, you're right, we have grown in depth. A few years ago, we really didn't have as much knowledge to be able to offer. It's sort of like what you were saying, we have to offer something and the other partners do too. And so we really weren't at that place, but as you've commented and observed, I think we're at a place now where we can certainly at least put our toe in the water and think. And so we've been brainstorming, but your suggestions for how that might work are most appreciated.
Can I add to that one? I think if there was some sort of workshop, a think tank, and also having the people who've been in your research, they want to help. You just talked about it, and they may have good feedback that you may not have thought about in terms of where to go next or how to bring it to the community. So I would encourage that. I have one other question. In terms of the sequela for all of the testing, and this went back to Brian's work as well, is it constant amendments, or do you have your, it is, or is it you put as much as you can in the protocol that you can think of, and then you have to go back in?
I think it's a combination of both. So we also do build into our protocols to do testing, sometimes halfway through the protocol to see if this observation is needed. So we can take away some things and edit the protocol. We can also add things in as needed from our insights. And so that kind of gives us the flexibility to do that. I think a lot of the protocol development is changing. So hopefully that will give us more flexibility to really look at those things that are kind of highlighted to add those as we need it.
Congratulate all of you. It was really wonderful to listen and hear how you're moving the science.
Thank you. Thank you. Yeah, and for me, participants consent when they enter the protocol for their samples and data to be used for other research purposes, and we purposefully build that into our consent so that it is allowable, and then it's pre-existing, de-identified, and can be moved on to other questions. And then also funnel into repository protocols. And we have standard language as well as exploratory aims where you don't have to say everything that allow for that. And we're happy to share that information.
And I would probably like to address just a few things. One is coming from an academic setting at the University of Tennessee. When I came here, it's the same experience here as it is in an academic setting. So if anyone's out there going, well, it's different because you're at NIH, it's not different. It's the same thing. So while you may be at the College of Nursing, you can also collaborate with the College of Medicine and use their basic science labs. It's the same thing here. We're just going to our neighbor and saying, hey, let's partner, let's share joint authorship. And I've got some resources, you've got some animals, let's throw them together and see what happens. So I want to make that point that it's not magic here. And in fact, I would say the IRB may be harder here, and some of those issues are being addressed. So that's one thing. We have vanishing subjects all the time. So, you write a protocol, you've got a lot of people out there with that disorder, they don't show up. So I can show you protocols who have under-enrolled. So while we're presenting well, and they're doing lots of things very well, but again, there's not anything that magic about that either. So it is always a struggle. It's always thinking about the feasibility of actually being able to do what you put in there. And we talk a lot about subject burden. And, okay, if you put another questionnaire in there and that adds another day, will they come back for that next visit or not? And so we're always balancing that. And I do think that Dr. Gilman made the point that the mission here is, I paraphrasing terribly, but it was along the line of, our mission is to provide hope. And so that's a piece of it. We provide the hope that maybe they'll be able to use what the results of these studies. But even if not, these are usually patients that come in that are more than happy to provide information for the next group of patients that are coming. And you can see that in the video that's on the Discovery Channel about the clinical center. There are many, many good examples there of that. Okay. If there are no other questions, is there anybody else in the audience that wanted to ask a question? Okay, if there are no other questions, then Dr. Grady, I will give this back to you.
And I want to add my words of commendation to the presenters and to the progress that's been made. I always get progress reports in between, but it's good for me to hear each year and be able to have a watermark for how much has changed. And I also would underscore your comments, Anne. I do think that the only thing I would say to add to that really is that each person that you're looking at here really is a pioneer in a way, because it's what Leo put his finger on when he said part of the idea of the collaboration is you have to go and explain to people what it is you do and why they should care and what you have to offer and trade back and forth. And I do think that this environment is probably very much like your environments, except it's almost an environment that didn't have a nursing school and has to figure out what it is because intramurally we didn't have as large a presence. And so from an intramural perspective, there is a lot of discussing what we're doing and why it's important, et cetera. It's almost as if I've been doing some visits again out and about to find out what's, sort of see what's happening in the outside world kind of thing. And someone commented that I don't know why people come to a university when they know what the philosophy and mission is and they want to do something else. Why don't they come, to a place that matches their mission? And so I was struck by that because it's quite sensible. And it's a very good question. But I was struck by the fact that in some sense, our intramural program are people who've come here to try to teach the organization a part of the mission that they didn't know they had before. And so I think that the progress is wonderful, but I do think that you all do deserve a lot of extra credit for the work that you've done. So with that, we are really coming to the end of our day here. We're finishing on time. I want you to notice that because this is a very complicated agenda this afternoon. But we've covered a lot. We've heard a lot of exciting new things that are going on. We have a better appreciation of what's happening at the clinical center and all of some of the drivers that do move us in a forward direction. We will adjourn this afternoon. You can leave all of your papers and anything you want to in this room. It will be locked, and we are meeting here in the morning. We will reconvene promptly tomorrow at 9 o'clock. And tomorrow will be a closed session. We will review the applications that have come before us for this council for the purpose of recommendations to move forward. So with that, thank you very much for your attention. We're happy that you made it here, and we'll see you in the morning. Have a good evening.